Hi, we're Tosh and Max, and we're AP Biology students sharing our final presentation on the PTC PCR lab. This lab helped us figure out whether we have a specific gene that allows us to taste a compound called PTC. To do this, we extracted DNA from our cheek cells and used a technique called polymerase chain reaction, or PCR, to amplify the TAS2R38 gene, which is linked to PTC tasting. After amplifying the gene, we used gel electrophoresis to separate the DNA fragments and visualize our genotypes. Based on the band patterns in the gel, we found that Max is a heterozygous genotype, uppercase T and lowercase t, and Tosh also has a heterozygous genotype.
Unit 1 focuses on the foundational chemistry that makes life possible. All biological molecules are built from a few key elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, often abbreviated as CHO and PS, or CHOMPS. DNA, the molecule we analyzed in our lab, is a nucleic acid made from monomers called nucleotides. Each nucleotide includes a sugar, phosphate group, and nitrogenous base, and these bases form hydrogen bonds that stabilize the double helix. In our PCR lab, we used heat to denature DNA, breaking these hydrogen bonds so that strands could separate. This is essential for the amplification process. Another key concept is the role of buffers, which we use to maintain optimal pH conditions during PCR, preserving enzyme activity. Finally, we used gel electrophoresis to separate DNA fragments based on their molecular charge and size, showing us whether our restriction digest successfully cut the TAS2R38 gene. All of these chemical interactions are grounded in the principles from this unit.
Unit 2 focuses on the organization and function of cells, the basic unit of life. Cells contain various structures or organelles that each have specialized roles, such as a nucleus holding DNA, the plasma membrane controlling what enters and exits, and ribosomes building proteins. Eukaryotic cells, like the cheek cells we sampled, are complex and compartmentalized, which allows them to carry out highly organized processes. In our PCR lab, we applied this knowledge by extracting genomic DNA from our own epithelial cells. We used a saline solution to collect the cells, then heated them to break open both the cell membrane and the nuclear envelope, releasing the DNA into the solution. Using a centrifuge, we separated cell debris from the supernatant, which contained the DNA. This process relied entirely on our understanding of cell structure, particularly how membranes can be disrupted and how cellular contents can be isolated. Without this foundation, we wouldn't be able to carry out such precise molecular biological work.
Unit 3 is all about how cells obtain, store, and use energy. The main concepts here include metabolism, enzyme function, ATP production, and the processes of cellular respiration and photosynthesis. Metabolism involves a balance of catabolic pathways, which break molecules down to release energy, and anabolic pathways, which build molecules using energy. Enzymes are proteins that speed up these chemical reactions by lowering activation energy, and ATP serves as the energy currency that powers cellular work. This unit connects to our PCR lab through the role of enzymes, specifically Taq polymerase. This heat-tolerant enzyme synthesizes new DNA strands during PCR, functioning optimally at high temperatures. The PCR process includes heating to denature DNA, cooling to allow primers to bind, and reheating for extension. Enzymatic activity here is directly influenced by temperature and pH, just as we learned in this unit. Though we didn't measure ATP directly, the energy flow concept still applies. Enzymes power the replication reactions just like in living cells.
Unit 4 explores how cells grow, replicate, and communicate. The cell cycle includes phases like interphase and mitosis, during which the cell duplicates its DNA and divides. Regulation of the cell cycle involves checkpoints and molecular signals that ensure cells only replicate when it's safe and appropriate. Cell communication, meanwhile, includes chemical signal pathways that coordinate responses from growth to immune defense. Our PCR lab mirrors parts of the cycle, specifically DNA replication. We use primers to target a specific region of DNA, the TAS2R38 gene, and copied it using thermal cycling instead of enzymes and checkpoints. Just as cells use molecular signals to regulate where DNA is copied, our PCR machine used programmed temperature changes to trigger each step of the reaction. Understanding cell communication and replication helps us grasp why PCR works so effectively and how it mimics what cells do naturally.
Unit 5 focuses on heredity, the transmission of genetic information from parent to offspring. This unit builds on the work of Gregor Mendel and his discovery of dominant and recessive alleles. Genes exist in different forms called alleles, and whether someone inherits one or two copies of a certain allele determines their genotype. The genotype, in turn, influences the phenotype, which is the observable trait. Tools like Punnett squares help predict the probability of inherited traits. In our lab, the ability to taste PTC in a trait controlled by the TAS2R38 gene and inherited in a Mendelian pattern. The dominant allele, uppercase T, allows people to taste PTC, while the recessive allele, lowercase t, does not. By tasting the paper, we predicted our phenotypes and then used PCR and restriction digest to analyze our actual phenotypes: homozygous dominant, heterozygous, or homozygous recessive. This gave us real-world example of how traits are passed down and expressed, which is exactly what Unit 5 is about.
Unit 6 explores how information in DNA is turned into proteins and how that process is controlled. The central dogma of molecular biology, DNA to RNA to proteins, highlights transcription and translation. But gene expression is not constant. It's tightly regulated by things like transcription factors in eukaryotes or operons in prokaryotes. Mutations or variations in DNA can lead to changes in the resulting protein or how it's expressed. In our lab, we studied the specific gene, the TAS2R38 gene, which determines whether a person can taste PTC. This gene is expressed into a receptor protein on the tongue. A person's ability to taste PTC depends on the version or allele of this gene they carry. The dominant allele results in a functional receptor. The recessive version does not. We amplified the gene using PCR and then used restriction enzymes to analyze the version present. This experiment showed how every single nucleotide differences can alter gene expression and affect traits.
Unit 7 covers the principles of evolution by natural selection, originally discovered by Charles Darwin. Organisms vary genetically, and these differences can affect survival and reproduction. If a trait improves fitness, it becomes more common over generations, a process called natural selection. Over time, this leads to changes in the genetic makeup of populations. Our lab connects to this unit through the TAS2R38 gene. The ability to taste bitter compounds like PTC might offer a selective advantage by helping people avoid toxic substances in the nature. However, the non-taster allele is still common, which suggests that in some environments, being a non-taster may also be beneficial, or at least neutral. By observing the variation in our own class and linking it to the real-world evolutionary pressures, we saw firsthand how genetic variation fuels evolution and how alleles are passed on or selected against.
Unit 8 focuses on the interactions between organisms and their environment. Traits that affect survival and reproduction, or fitness, are subject to natural selection, which in turn impacts populations and ecosystems. Ecology explores how populations evolve, how traits affect relationships in food webs, and how environments shape the organisms within them. The TAS2R38 gene from our lab provides a lens for exploring genetic variation within populations. Even though tasting PTC may seem like a minor trait, it could influence what foods a person avoids, which can impact nutrition, survival, or toxin exposure in specific environments. Interestingly, both the taster and non-taster alleles are present in modern populations, which raises an ecological question about balancing selections or trade-offs in different environments. This lab gave us a glimpse into how genetic and ecology intersect.
My bioethics slide focuses on the ethical debate around pre-implantation genetic diagnosis, or PGD. PGD is a process that happens before pregnancy during the vitro fertilization. It allows doctors to test embryos for genetic disorders and then select which ones to implant. Essentially, it lets families avoid passing on serious inherited conditions by identifying and choosing embryos without those genes. Supporters of PGD argue that it prevents a lot of pain and suffering. By screening for serious diseases, family can have children without life-threatening genetic conditions. They also say that PGD is a medical advancement that improves reproductive freedom and helps parents plan for healthier families. On the other side, critics raise ethical concerns. They worry that PGD could lead to designer babies, where parents choose traits like eye color or height. More importantly, some believe that it devalues human life by discarding embryos that don't meet certain criteria. This raises major concerns for people of faith. From a Christian or Catholic perspective, life is sacred from the moment of conception. That means every embryo is considered a human life with dignity, regardless of health or traits. So using PGD to discard embryos is seen by many religious groups as morally wrong. This topic brings up hard questions about suffering, control, and the value of human life, and it shows how science and faith can sometimes come into tension.
CRISPR is a powerful gene editing tool that allows us to change DNA to prevent genetic diseases or conditions, offering the potential to reduce suffering and save lives. Supporters see it as a breakthrough that not only treats illnesses, but also gives parents the ability to protect future generations from inherited disorders. However, critics raise serious concerns. Many of the long-term effects, especially on children born from edited embryos, remain unknown. There's also the fear that CRISPR could deepen social inequality, with access limited to the wealthy, creating a divide between those who can afford genetic advantages and those who cannot. On a deeper ethical level, some argue that CRISPR-Cas9 challenges the belief that every person is made uniquely in the image of God, raising the question, by editing our genes, are we interfering with something sacred?